protocol today, we're going to focus mostly on medical devices. You guys will get a copy of this. What we're going to talk about today, we're going to first just discuss what is a study protocol. We're going to look at some of the key pieces when developing a study protocol. So who's your audience, who are your key people, where's your research location plays into it. Some of the do's and don'ts of writing in general and writing a protocol. Some of the items that you need to consider when looking at developing a study protocol, the key elements of a protocol. Then we're going to go through some of those key elements, not all of them, otherwise this would be a very long session. We're going to discuss how the protocol leads into your other essential documents for clinical trials, and then we're going to conclude, and then we'll do a little in-person activity for the people that are here. So what is a study protocol? Per GCP, ICH guidelines, it's a document that describes the background, the rationale, the objectives, the design, the methods you're going to do in order to achieve answering a research question, right? But what it really is, it's a legal document and it's a study plan, and it's all designed to answer a key question. So it all starts with a question. And your protocol is really kind of like your recipe. And it's part of a larger program. So it's important to keep the big picture in mind. So when you're dealing with devices, let's say this is your first study you're doing and it's your first iteration. Hi, no problem, come on. It's your first iteration of trying to collect some data. Still keep the big picture in mind. Understand where is this program going as a whole, and what can I incorporate into this preliminary study in order to achieve some later goals or answer some later potential questions. So think about your big picture, and you can design your protocol in a way that can answer multiple questions at the same time. You could be testing a hypothesis, getting some preliminary data, collecting data for a submission. I don't know why it's moving, but sorry. Collecting some data for a submission, to get a product to the market, or to prove equivalence to a current standard, right? So thinking about who your audience is probably one of the key first steps. When you write your protocol, you want to think about, who's your audience? So are they individuals within your organization? Because if they're individuals within your organization, they're going to be interested in answering questions related to the product, right? Are they individuals outside of your organization? Are they scientists? Are they researchers? The IRB will be part of your audience because you need to get IRB approval for any clinical study that's involving human subjects. You will have to deal with monitors, so they're part of your audience. Is it the FDA, right? So when you're dealing with the FDA, you have to think about which office of the FDA am I dealing with, and design your protocol in order to address the questions that these key audience members will have, right? So another piece of this is trying to, when you think about your audience, is try to involve those key members at an early enough stage that they can give you that feedback that you need, the questions that they'll come up with earlier on, so you can design your protocol with them in mind. So if we're talking about the IRB, they're going to be concerned about safety of subjects, right? You're going to have a really good device description. You're going to have a nice risk analysis laid out in your protocol plan. Right, in a way that regardless of who the audience member is that's reading your protocol, they have the same takeaway, right? So when you reread what you've written, make sure you put them in your mind and think about, what is this person taking away from this protocol? So for every piece, keep the big picture in mind. What's the end game? What's the program plan as a whole? This might be a preliminary version of your device that you're testing, but you want to design for potential future versions that you're aware are going to be coming down the line, and explain how this study plays into the big picture goals, right? So this really limits the amount of changes you then end up having to go back and do to a protocol. And most protocols, you do have to go back and amend and make edits, and that's fine, but that's all time on your end, right? So you want to think about going forward, how to design in a way that's going to think about all those pieces. So who should be involved, right? think about everyone involved at every step of the protocol, whether it's in the development of the protocol steps, study conduct, is it your PI, principal investigators, study staff, nurses, doctors on site, the people who are going to be doing the data analysis for you. And during protocol development, figure out at what time points are these the right people to involve. So if you're doing research with an academic institution, we're right by UCI, if you're doing it with UCI Medical Center, you're going to have to think, do I need their site staff involved? Yeah, probably. Do I need to contact biosafety? Do I have blood samples I'm dealing with? Do I need to contact radiation, right? Their medical staff that you'll be dealing with, hospital administrators, a lot of times they have their own process too that you have to go through. And they have, a lot of times academic institutions have their own IRBs. Are you dealing with a CRO? Do you get to wash your hands of everything if you're dealing with a CRO? Or are there pieces that are your responsibility? In the end, if things don't get done the way you want them to get done, you're left with the end result. So be involved early on in the process so you're able to mitigate some of the risk you're taking on, right? So before the study starts, think about who and when these key individuals need to be pulled in, right? And you'll always have your sponsor, your investigator. I don't know why this is running through everything. Sorry guys, I'm just gonna do the old fashioned way. Think about who your sponsor is and who your investigator is and who these key, no, this is where I want to be. And who these key people are that are involved in the study, right? But don't assume that they understand their role. Because in one study, the sponsor can take on a subset of responsibilities. In the next study, they may divide up responsibilities between them and the investigator, right? So you want to have very clear instructions and a framework of what responsibilities fall on who, okay?
Want to try this instead?
Sure. Let's see, there we go. Okay, so think, and then also think about where the research is being done, involve all those key players. Are you at an academic institution? Are you at a private practice? So if you're at a private practice, they may not have the support there available to you for a clinical study, right? So you have to make sure all of those pieces are in place. As the sponsor, the responsibility falls on you, right? Are you having this research done by a CRO? Have you qualified the CRO? Do you know of other research they've done? Academic and non-academic, you fall into a different category, right? Academic institutions and major hospitals a lot of times have their own IRBs, but it doesn't mean that that you'll be using just one IRB. You may be using an external IRB for the ethics piece of things. And their internal IRB may be responsible just for making sure that you've adhered to that institution's rules and regulations around conducting clinical studies. So make sure you have something in place, a document in place. A lot of times it's called a reliance agreement that divvies up responsibilities among the IRBs. Because on monitoring at the end of the study, what ends up happening is if you have two parties that are responsible for the same activities, if something goes wrong, what happens? Everyone points their fingers at the other guy, right? So you need to have very well ironed out responsibilities among the IRBs. I'm going to jump into some of the do's and don'ts of writing, and then we can jump into the protocol pieces, okay? This goes for all writing, but specifically protocols in this case. Be consistent in the message. Make sure that you've ironed out your research question that you're trying to answer, and that when you're writing your protocol, it flows to address that question. Understand what information goes where. Look up templates of other protocols, right? In one protocol, it might be called statistical analysis, and another protocol might be called a statistical plan. There are multiple names for the same things, but understand what information goes under which subheader. Look up templates that are relevant to your device. So if you're dealing with a class 1, class 2, class 3 device, you're most likely going to have a predicate out there, right? Clinicaltrials.gov is a wealth of information. If you know what your predicate is, you can look up what your predicate's company did for their studies, their clinical studies, in order to support their products. Try to state information once. This is such a huge pitfall. When you end up stating information multiple places within the same protocol, what ends up happening inevitably is at a later date, you need to amend the protocol, make a change, and start doing the study slightly differently. And then you end up with contradicting information in the same exact protocol. Do not expect that your reviewers and your IRB and your PI will catch that. Because you're the only person that's that intimately familiar with your product and the study you're designing. They may think, hey, it was intended to be this way, right? I always say this, write your objectives first and make them comprehensive and specific. Be as specific as possible in writing your objectives. Your objectives will basically guide you through the writing of the remainder of the protocol. Even if you have another person writing the protocol for you, as the person responsible for the device and the study itself, write the objectives, or at least be very involved in the objective writing so that it's a very clear message, the message you're giving, right? And try to plan up front so that you're not stuck with too many amendments. Multiple amendments to protocols and consents end up causing multiple issues when doing data collection down the line, right? One PI or one of your sites will think, hey, we're still working with revision A. Your other site will say, oh no, we're on revision B already. And so you have two different sites that are part of the same study that are doing it differently. Some of just the basic considerations, think about the big picture, right? With your product, where is it going? What is your plan for your product, your device for the next 5 years? Is there anything you can implement in this initial study to help support a regulatory submission down the line? Is there a usability piece that you can incorporate? These are all ways to be really efficient in developing your protocol. And it all has to do with thinking about the trajectories of the company. And that doesn't mean you have to put it all into one place. But if something's on the horizon and it can be achievable immediately, then take advantage of what you have. Think about your study population. What's feasible within that study population? Are you dealing with wound care? Are you dealing with diabetics? Are you dealing with people who are going in for surgery? Think about all of those pieces when designing your protocol. And then who'll be helping conduct the study? And by that, I mean the day-to-day activities of conducting the study. Is the trial a primary part of that PI's function? So if you're dealing with a physician's office and patients are coming in for, let's say, a pre-op procedure, can your trial involve 20 steps? Can your study involve 20 additional steps for them? No, because you're really a secondary function of what their day-to-day activities are, right? So that's almost guaranteeing that something's going to go wrong or something's not going to get collected properly, right? Or do you say, hey, yes, I need these 20 steps. You don't have to collect it on every single patient who walks in the room. Collect this information on your last patient of the day so that I know that you don't have anything waiting for you on the other end and we have the additional time to do this, okay? And then what's a reasonable length for a study? Overall and per subject. And that's when looking at your protocol development team is really crucial. So you might design this great study that gives you a ton of information, but a principal investigator who's done a similar study before, or a mentor, or a research scientist, or a clinical research associate might come back and say, hey, I saw this done this way previously. It's not going to give you as much information, but you're achieving the same goal and you may get more subject enrollment out of it. So you kind of have to play with the protocol and involve the key people that could give you insights into what will be helpful. So back to the protocol. When we're dealing with a commercial product, our basic objective, right, is to get the product to market, right? You want to prove that it's safe and effective, it meets a standard, and you're validating the performance, whether it's against a current standard or validating the performance is repeatable, right? With drugs, we're dealing with phase 1, phase 2, phase 3 studies. With devices, we're dealing with pilot, pivotal, and post-market studies. Think through the objective of the study and write it very clearly, very well-defined study objectives. Everything flows from the objective, right? You're trying to answer a question. What is the question you're trying to answer? An unclear objective is something like, hey, I want to evaluate safety and efficacy. But what is that really telling me? Not much, right? And it's not telling your reader very much either. But if you write down, my study objective is to collect data from this subject population, let's say a dialysis patient population, and I'm going to use this device, and I'm going to run this study at dialysis clinics, right? And I'm going to compare my device readings to another standard of care. Might be a blood draw, might be et cetera, right? When you write a clear objective, it starts to write your step-by-step procedures for you. Okay? Be specific. You need to get what you need. You need to get the information to answer that question out of the study, right? So be as specific as possible. Consult with the right people, pull your interdisciplinary team together, involve a statistician. This is key so that you understand what statistical significance is for your study, right? And involve a statistician that's dealt with clinical studies before. Because you'll get very different answers from a statistician that has not dealt with clinical studies before. Pull on the clinicaltrials.gov information out there. If you see that your predicate did 20 subjects, go to your statistician and say, hey, is 20 subjects significant for the study? They might come back and say, no, you only need 10. And so you could start in your protocol and say a minimum of 10. But let's say you expand out and you get to 15, it's still an option for you. Then pull together a protocol synopsis. This is basically a summary. It's going to include your title, product details, right? Be very detailed in your product description. Nobody else knows your product but you. Clinical endpoints, you can have multiples. You don't have to have one clinical endpoint, right? You can have a primary and secondary clinical endpoints. What are you comparing to? What standards out there are you comparing to? A general overview of your study plan. So that the reader and anyone you consult with understands what you're doing in order to achieve a goal, right? And then stop at this point and review with your team, right? Get everybody involved from your interdisciplinary team at this point. Do not go through and write a 30-page protocol and create an entire plan when you don't have everybody kind of on the same page, right? So these are the elements of a protocol. What I'll tell you is they'll be called different things in different protocols, but an overview of elements are contained within this page, and you'll get a copy of this. You'll have to have background information, your objectives, which you've already written first, statistical methods like statistical analysis plan, and introduction. This will have a brief description about your technology. You will be submitting additional supporting documentation to your sites. Study rationale, study design, schedule of assessments. We'll discuss subject enrollment. Who are your subjects going to be? What is an inclusion criteria? What is an exclusion criteria? What are you going to do in the case of subject discontinuation? Somebody decides to drop out midway through your study or move states, right? A really thorough device overview. Your PI needs this, the IRB needs this. Anybody referring to your documentation for your study will need this. What are the procedures related to the study? So if you're just simply measuring something that is a consequence of like a surgery, is that a study procedure? No. The study procedure is measuring it, right? So make sure to clearly define what are your study-related procedures and at what point are those study-related procedures going to take place. How are you going to deal with adverse events and serious adverse events? What do you expect to have as an adverse event from your device or product? How's data going to be handled? How are you going to QA the data? How are you going to monitor the data? Where is your data coming from? Is it coming from the health record? What's your source? So yes, the health record may have I don't know, the potassium of a specific patient. But is that the source? The source is what came from the blood analyzer, right? So that's what you're comparing to. What are you going to do about protocol violations? Does it automatically mean you discard that subject, or specific protocol violations cause you to discard subjects? If it affects your data, right? So there's no reason to discard the subject overall. What are your stopping rules? You need to have your ethical regulatory considerations, right? Who's your IRB? How are they going to review? What's going to be your process? You're going to have a reference section. Sometimes when people write their first protocol, they'll put like 100 references in there. 20 is a good ballpark number. Okay. Nobody's going to go looking up your references. Put the most relevant information in there, right? Information that you probably pulled from to create your background and introduction section, right? A list of abbreviations. Everybody has their lingo, right? So in clinical research, where we throw around words like CRF, CRO, blah, 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 and every company has their own lingo. Have a list of abbreviations, state it once, you don't have to repeat it throughout the whole protocol, right? And then a supporting document list. Okay.
What are the supporting documents?
You can discuss your other documents related to the study, your consent, your CRF, etc. You can, if you have an investigator's brochure and you have more details about your device, you can put that in there. You know, it doesn't, the protocol is not a dumping ground for everything. So it's good to compartmentalize things into separate documents if needed, okay? Now, some protocols are very simple, right? I'm gonna give subjects a questionnaire and ask them about their daily activities, what they're eating and what their exercise is. I don't think those are gonna have very many supporting documents. But when you're dealing with medical devices, they do have quite a bit of supporting documentation, okay? And of course, an implantable will have more than a non-invasive device. So you kind of have a reference. Background. This defines the product. It mentions the disease or the condition where this product specifically can be of help. Why are you using it with this population? It provides a summary of the data, the background data, so clinical and non-clinical. So if you've done some clinical, or there's clinical out there that supports why this specific device could work in this population, pull some of that into the background. That's great supportive material. And there's also non-clinical, right? We all start with bench testing, animal testing, use that to support your device, right? Study rationale. So this is really, it's answering the question of why and what. Why this research needs to be done, what is its relevance? And it's brief. It's a brief description of some of the studies that are out there that are published that say, hey, this device could potentially work in this way, in this population, right? And these are the studies that support them. It also discusses the relevance of studying this device in this specific population, meaning that, hey, I could study it in a general population, but it's this disease state that this could help with. So that's why I'm studying it with this specific subpopulation. You want to do a risk-benefit assessment here. So the risk of running the study, right, the risk to the subject of participating in using this device versus a general benefit, either to the subject or to society as a whole. You do not need to have a benefit. You just need to justify that the risk is acceptable within the context of the study.
That's the risk of using these subjects for that protocol, not the risk of the actual device.
It is the risk of the device as well.
Always,
Yeah, so it's a risk benefit, right? So you can say there is a risk associated with putting this LED-based technology on my fingertip, right? It can risk what? Sensor burn, et cetera, et cetera. But the benefit might be that I am now getting non-invasive SaO2 or SpO2, right? Which is, for society as a whole, a huge benefit, right? Because then you're not having to take a blood draw from an artery to get that same metric, right? So you have to justify within the big picture why this study makes sense to do, okay? That's also why we involve statisticians. As a product developer or a device company, you might say, hey, I'd love to have 100 subjects involved. But the IRB might come back and say, wait, why do you need to do this to 100 subjects? What is your statistically significant sample size? Let's start there. Prove, because within your study, you're also going to prove that this is a true risk. We've done 20 subjects and nobody's had an issue, or one person's had an issue, and sensor burn was not one of them. Maybe the adhesive irritated somebody's skin. So now you know that's my AE, not sensor burn, right? So it gives you information. And then later on, you could expand out and do a larger study if you're doing it for the purpose of marketing or publication. Okay. So your study objective is really a statement of the research questions. What are you trying to answer here? Be simple, be specific. I think I've reiterated this multiple times, but this will really write the rest of the protocol for you. Your desired research outcome is your endpoints, your study endpoints. So this might sound familiar. I think you guys have a resident that's doing something like this here. Let's say your objective is to compare 2 methods for the treatment of diabetic foot ulcers, right? You have 20 control and 20 test subjects. And with the test subjects, you want to use a new method. And what you're measuring on the other end is, does it help to reduce the time it takes for them to heal, right? And does it increase the closure of the wound? So you could be physically measuring the size of a wound, the surface area of a wound. And you want to collect this data to answer the research question. So your primary endpoint, you need your measures, what are your outcomes, and it'll answer your primary question in the protocol, right? So look at it as a big picture. Include your study endpoints within your objectives. This is what I want to collect in order to achieve answering this question. Okay, and you can have multiple endpoints, right? Because you can say, I want to see if it's improving wound care with these, the state of these wounds, but I also want to understand, is it improving overall lifestyle for these patients, right? That's another endpoint. Study design, and this should really flow from your objectives as well. It tells you how the study is going to be designed. So think about the screening time period. Let's say you want to screen these subjects ahead of time. Think about the types of controls that you'll be using. What are you comparing to, right? Is it a standardized control across multiple sites, right? A randomization plan if you need one. Types, time periods, and the duration of the study as a whole, right? So let's go back to the diabetic foot ulcers for a second. Let's say most physicians' offices say it takes about 12 weeks for one of these to heal. Otherwise, we have to use an alternate treatment. Does it make sense for you to say, oh, wait, I want my study to be 36 weeks long? No. Does it make sense for you to say, I want my study to be 6 weeks long? No, you won't see the change, right? And then jump into the procedures where you're giving more details, like the step-by-step details. Procedures and methods, right? This is how I'm going to collect my data for the study. This is what I'm going to do from start to finish. There are detailed directions and descriptions of how you'll be running the study. Include the relevant clinical data that you'll be collecting, right? So if you're collecting demographics, include that in your procedures. If you care about the skin tone of a subject, you're going to use a scale, like a Massey scale, to evaluate skin tone. Make sure you say, I'm going to evaluate skin tone using a Massey scale on every subject. I'm going to collect vitals. Don't leave it general. Tell me what vitals you're going to collect. Blood pressure, heart rate, height, weight. It can even be as simple as, I want to collect the size of a finger, because that's where my device goes. It goes on a finger. I want to measure the circumference around the finger where my device is going to be placed. That's significant. Put it in there. Medical history. What part of the medical history is relevant for your study? What part of a physical exam is relevant? And if you need to, include a schematic. A lot of times pictures really help simplify the process for the person interpreting your protocol on the other end. Namely,
the protocol.
Exactly, namely the PI. I also like to include a schematic or a picture when doing your device description. You can talk about a device all day and all night, but until somebody holds it and sees a picture of it, they have no clue what it is, how it works, where it goes, right? Describe your control and your test population. all of this will reduce the confusion that comes from running a protocol, like the day-to-day activities. Who's your control population? Who's your test population? You don't have to put all of this info into the methodology. You can have a study manual, and that can have more information in there, right? So give an overview in the methodology. Make sure to highlight any key pieces you're going to collect. Nobody's going to collect heart rate for you if you didn't say that you wanted the heart rate in the protocol. Okay, so put the key pieces. You might come back and say, hey, I might collect heart rate using a pulse oximeter or an EKG, not necessarily, give yourself options. Okay. Next is your schedule of assessments, and your methodology really leads you into your schedule of assessments. This is the sequentially planned protocol events, right? So patient walks in on day one, I'm going to consent them on day one. I'm going to collect their general demographics. I also want to collect weight, height, skin tone, heart rate, and blood pressure on day one. Make sure that this is super easy to follow. Put it in a table, right? Timeline on the top, on the left-hand column, any events or activities that are going to be taking place. If there are clinical events that normally happen, put those into your schedule of assessments. So if you're dealing with a principal investigator that's a surgeon, say, hey, I want you to weigh them 2 weeks after the surgery. I want another weight at that time point. This then feeds into your case report form, which is basically where you're going to get a lot of your data post-study. Okay. And this is kind of a example schedule of assessments, right? The top is the timeline. The left-hand column are all the activities. So you'll notice you have demographics, randomization. When are these activities going to take place? If you look at something like weight, I'm collecting it 6 different times over the length of the study. So use X's or check marks to kind of show schematically when are these activities going to take place. It makes it much simpler for somebody within the practice or within the clinic who's helping you collect your data to flip through to the schedule of assessments and say, wait, this is visit 3. What am I doing today? Right? Because they're doing their regular day-to-day job. They need a quick reference guide. Study enrollment. This is your inclusion exclusion criteria. This basically tells us what subjects are going to be participating in the study and what subjects should not participate in the study, right? What conditions are contraindicated for with this device. So if you tell me that I'm placing an adhesive sensor on someone and I know that my adhesive is the same adhesive that's used in Band-Aid bandages, your exclusion criteria should be, are you allergic to adhesive on bandages, right? Is there a certain population that's unsafe to participate in the study? Let's say somebody with a certain cardiac condition, or not unsafe, but will not give you valuable data, right? So if they're not going to give you good data and they're not going to give you data that you value, omit them from the study, put them underneath your exclusion criteria. If you're not using the device on the intended population, explain why you're not using the device on the intended population. So let's say your device is designed for children with a certain condition, but it's hard to do studies on children, right? So you're going to start and do your preliminary study on adults that have the same condition, or healthy adults in a way that mimics that condition, right? Explain that in your protocol when underneath your inclusion exclusion criteria. Think about minimizing the risk, right? Reducing the number of adverse events that could occur. So this all leads into inclusion, exclusion. It really iron it out so that it's a very clear cut. So there's no confusion when subjects come into a room. It's then going to be referenced by the PI and their staff when deciding who can participate in the study, right? So they automatically know, hey, this device is LED based and it goes on the wrist. Oh, they say that they don't want anybody with a scar over the site where the LED goes. Okay, that makes sense. So you're not in the study anymore, right? Think about your randomization, your blinding. Are you going to be single blind, double blind, no blinding at all? Not necessary to have blinding, but have a plan in place.
What do you mean by single and double blind?
So single blind is if one part of the study is blinded, we don't know what they got. It's a lot of drug studies are like this, you know. I don't know what pill you got. We'll find out after the study.
Oh, you don't know which one is
Concludes.
placebo, which one is not,
Exactly.
okay.
Double blind is it's a double uncover. So it's not so relevant for device studies. A lot of times device studies, it's kind of like, I'm using the device, I'm not using the device, so it's not blinded, you know?
Yeah.
That's why I'm saying it's not always necessary to have blinding. And in some cases, you might be using, like, the current standard of care is a diabetic drug, and then you have a new diabetic drug you're trying out, right? It's not ethical to not give a diabetic person the drug they need, right? So you have to have some kind of standard that you're giving. Do you get what I'm saying? Or it's like cancer drugs.
Mhm.
It's a new cancer drug or the old one that they were using. You can try the new one. And this is this group that's trying the new one. And this is the older group that's trying
The old one.
the old one. But you can't say to a
Not
cancer patient, I'm giving you placebo.
Oh, yeah,
Right?
yeah.
Right. So subject withdrawal. This is so important because you
Yeah.
cannot decide once they withdraw what you're going to do to enroll another subject. It must be in the protocol up front. If you have a dropout or you have a reason to get rid of a specific subject, and there's always reasons to get rid of subjects. Let's say you have a non-compliant subject. Someone who shows up once every 5 visits, you need them.
It's pretty like expected
Yeah.
to happen.
Yeah, exactly. So have a plan in place. Say, hey, I'm gonna try to follow up with the subject 3 separate times. If I don't get a response, I'm going to enroll another subject.
Can you explain one more time the double plan, single brand?
Can we get back to it at the very
Okay.
end? Sorry, it's just, there's quite a few
I'm sorry.
slides left.
Okay,
Okay, sorry. Okay, sorry. Adverse event reporting. So this is important to think about in relationship to your device, right? Like, what does it do? If you have a sensor with a spring on it, right? Could an adverse event be to indent the tip of the finger? Yeah. Because if you don't put it in there, then it's an unanticipated adverse event, which automatically gets reported. Huh?
On slide 18.
Okay.
Oh, I see.
So list all of your expected and anticipated adverse events. So an adverse event is an untoward medical occurrence that could be associated with a device or a drug, right, in a human study. So define what's expected and anticipated versus unanticipated.
What happens when you get an unanticipated event?
It has to be reported to the IRB within 5 days, and specific serious unanticipated adverse events also have to be reported to the FDA. So there's a whole set of regulations around adverse event reporting. And you can find those in 21 CFR 812. This information is out there. Do not expect the PI to be aware of it. Make sure that it is in your protocol, right? So that you have a clear set of rules and you say, hey, if these things happen, I as sponsor want to be notified within 2 days. Okay, you don't have to notify the IRB because I said they're anticipated, but I want to be made aware of it, right? So if you find out 3 days into your study that every single person you put your adhesive sensor on is getting a reaction to the adhesive, the PI shouldn't continue with the study. They should notify you so you can do something about it. Decide to pause the study, decide to
Redesign.
yeah, exactly, redesign. Or, hey, is he enrolling people who are allergic to Band-Aid bandages? You know, it could be that simple. And unanticipated adverse events are serious effects that could be related to the device or drug that we didn't expect.
So what's the timeline here? We need to report immediately or 1 day?
Depends on the severity of the adverse events, right? So that's why I put the references in there. There's a subset. A lot of times it depends on the class of your device, how severe the adverse event can really be. Some things are easily traceable to another activity that's happening in the study. Let's say somebody gets a bruise on their AC, right? And part of your study was to take a blood draw. The bruise is probably from the blood draw, not your sensor that's sitting on the hand, right? But that's why you write these details in there, right? Expect bruising from a blood draw. expect a skin tear or irritation potentially from the sensor. Those are all key things to have included in your adverse events, right? Make sure your protocol has clear information on what is to be done in the case of anticipated versus unanticipated AEs, including serious adverse events. Unanticipated is serious, right? Because you didn't know, you need to be made aware.
Because by then you've covered all your bases, and if something comes up, then that's.
Then it comes up and you
Has
go
to be,
about,
yeah.
exactly, it has to be addressed. So it's very important for your investigator to understand this, because you as the sponsor will not be aware of these things unless the investigator makes you aware of them. But the responsibility to report will fall on both parties, right? So they have to tell you, and then you have to say, yes, we have to report this within this timeline. And this is where we have to report it to. Okay. Statistical considerations and analysis. So what are you going to do with your data? Plan data sets to be analyzed. Which subject data will be included in each analysis? What's going to be randomized and what points are going to be used? So let's say you have a subject that you collect data on, and then you notice that you have really poor signal for 20 minutes in the center of your data set. Is that still data you want to analyze? Or what if you've noticed a lot of movement and part of your procedure is that they stay perfectly still while using your device, and you're getting funny readings during that time point? So you're going to have to include the exclusion criteria for your data points up front. Because if you don't, and then you go back and say, hey, you can't cherry pick data, right? You can't say, hey, this is good data, I'll keep this. And this is not so good, but I don't know what happened, I'm not going to keep this. You have to decide up front what is good data and what is bad data, right? Add in information about demographics that are going to be used to analyze the data. So if you're going to have a breakdown of males to females, and you're going to say I'm going to have around 40 to 60% males and females split, and that's acceptable, then put that in your data analysis plan, your statistical plan, because that's part of what the PI will also use. And in certain situations, the FDA requires it. So when you're evaluating pulse oximeter, they require that the data is not all skewed to males or all skewed to females. They require a breakdown of skin tones, right? Because light absorbs differently on different skin tones, right? They want to know that you have a range of ages, right? These are all details that they want when you're doing your data analysis. And say how you're going to analyze your data. What are you going to do? Are you going to do a t-test? Does a t-test make sense in this case? What did your predicate device do? Go look it up, right? Their submissions are out there. You can look up your K numbers, right, on predicate devices. You can look up their protocols on clinicaltrials.gov. Use that information. That's the value of finding the right predicate device. Even if it's not the exact type of device, if it's similar and it functions similarly, you could justify what you did to the FDA using that information. Okay. Make sure you have outlined how you're going to analyze your primary endpoints and your secondary endpoints, even if your secondary endpoint is as simple as there's an improvement in overall lifestyle. How are you going to analyze that? Do you have a questionnaire? Or are you just going to assume based on feedback that you get from their doctor? Even if that's the case, put it in there. Say how you're going to analyze it. What are your expected AE rates? So I'm sure you've all tested your devices in your labs, right? What would you, how much sensor burn would you expect? How much skin tearing or skin irritation due to an adhesive would you expect? Put information in there about an interim analysis. So let's say you have a 4-week study, but at the 2-week mark, you want to take a week break and you want to analyze all the data from the first 2 weeks. Then you want to restart the study. Put that information in there. That's relevant, right? So if at the 2-week mark, you realize that you have 6 males and 1 female in your study, then you need to go back to your PI and say, hey, I said I wanted an even split between males and females. Please enroll more females now, right? This is all relevant information. Data handling and monitoring. So data integrity is a big part of a study, right? provide information on how the data that's collected is going to be managed, how it's going to be coded for, how it's going to be de-identified as subject information on it. Where's the legend going to be housed? Right? Where does the subject ID come in and where does the patient ID and patient information go out? How are you going to monitor the data and verify for it? Are you going to take physical records from the lab? If you're dealing with a blood analyzer, do you have a human being on the other end of the study site writing down a number off of the blood analyzer? Are you getting a physical printout? Are you getting a download from the blood analyzer? Because the way you need to monitor for the data integrity is very different, and it depends on how you're getting that information. So if they're going and writing a hemoglobin value in a case report form, what are the chances they're going to make a mistake? Now you need to be able to look at your source. So you need all of these pieces of information, and how are you going to handle this data and monitor for its integrity at the end, right? What are you going to do about archiving data? How long can you hold this data for? Retaining records, right? What is going to be your monitoring plan for your study overall? How are you going to deal with confidentiality? You can say, hey, the site is going to handle de-identifying the patient information. But then how do you validate that what they're giving you is the right data for that right patient? So you have to think about all of those pieces.
How?
So you get a monitor.
No, how would you do that?
It depends on what they're doing, right? So you would hire a monitor, a clinical monitor, that'll go in and be able to look at the de-identified. You can also have the site do a double check on data that's transcribed. So let's say they get data from the blood analyzer or the health history, right? And it's decoded, they print it, they black out the patient information, they put the subject ID in there instead. Now you still have the original record from the health history. And you can have another person at the site who also has access to that information, validate that it's the correct patient that was put into the study, right? And that reduces the amount of errors that you're going to see. But when they're collecting 50 subjects in a day, you have to have these cross checks, right? Or else you'll get stuff you can't use on the other end. Ethical and regulatory considerations. So here, this is where you're going to talk about making sure you adhere to your GCP guidelines, right? And your regulations. In your protocol, you're going to have statements like this. Research will be conducted in accordance with blank, right? Whatever the regs are. So you're going to list all of your Declaration of Helsinki, et cetera, et cetera. All the stuff from my first PowerPoint about ethics is going to go in this section. Study staff will be trained to X, right? So there's training documentation out there that'll help you make sure that your staff understands what it is to be GCP compliant, right? There's city training, there's the other one starts with a B. Most academic institutions have their own training, et cetera. But you need, as a sponsor, you need to make sure that the site you're using has training records to good clinical practices, right? Add details on the study document on the ethics committee, which IRB is going to be doing what? What are you going to do in the case of an amendment? If my protocol is amended, it will be approved by the IRB prior to me implementing any of those amendments. We will halt the study, get another IRB approval, and then implement, right? Discuss what's going to happen with publications. This is so important to do up front. Who owns your data? Do you own your data? Do they own your data? Right? When you're dealing with an academic institution, it's important to think about those pieces. Who's the sponsor? And if you're the sponsor, do you retain the right to review anything they want to publish and approve it prior to their publication, right? Create a separate agreement that's for publication specifically. Put in there any financial disclosures that you need for the clinical investigator. If the clinical investigator is getting paid a fee, then it's the subject's right to know that this person has a vested interest in the study, right? Think about what you're going to do in the case of protocol deviations. What is a protocol deviation related to ethics, right? Someone didn't sign a consent. That's a big no. They didn't consent to the study and you collected data from them. Versus I misrecorded something. I took an inch off I forgot to record height and we asked for height, right? Can that be mitigated? Can you say, hey, this is a deviation, but the next time you come in, can I record your height? Yeah. Right, so there's solutions to the smaller ones. The clinical protocol really feeds into everything else, right? You have your clinical protocol, it's going to help you design your consent form and your subject information. It's going to help you design your investigator's brochure, your clinical study reports, your case report forms, and all the other documents related to having a clinical study that works together, right? That all the documents work together. Conclusion, when you're designing it, think about your team as a whole. Who are the key players you need? If you don't have them within your company, find them, right? Find your regulatory person, make sure they're involved early on. Define your objectives and your endpoints. Make sure you have well-defined roles and responsibilities. Compartmentalize the protocol. It's okay to write a protocol piecemeal. Hey, I'm gonna write my objectives, I'm gonna write my methods, I'm gonna write my references, then I'm gonna go and write down my ethical considerations, my stats plan, then pull it back together. But at the very end, when you read through it, make sure that you get the same message and it's a cohesive message. And it's all laid out to answer the questions you're trying to answer, right, in your objectives. Success in a protocol really depends on planning for the possibilities. What happens if there's a 2-week break, a 2-week Christmas break at a clinic midway through your study? So put those little pieces in there. What happens if subjects drop out? All of that should be included. Make sure the details are in there to answer your question. So if you read a protocol and you get one conclusion from it, you understand what it's pursuing, give it to somebody else and make sure that they get the same take-home message from it.